Before positioning medical products on the market, manufacturers have to be aware of product-related risk potentials regarding third parties, such as physicians and patients. Due to complex biotechnological architecture of diagnostic systems or sophisticated manufacturing processes for therapeutic agents, risks are always present that shall be under control of the manufacturer. Under control does not mean complete removal of any risk, but appropriate identification, evaluation, and minimization of risks lead to remaining risk that is under control. A prominent business model for identification, evaluation, and minimization of risks is made up by Failure Mode Effects Analysis, FMEA. This model contains tools to determine method reliability and detect weaknesses. Its importance is found within quality management systems to prevent and avoid mistakes. Risks primarily refer to patients, users, or the environment. According to these guidelines, risks consist of three aspects. First, the probability of damage happening, the severity of this damage, and the discoverability of this damage. Based on these three aspects, manufacturers have to declare safety of medical products by identifying known and predictable hazards. Quantitative evaluation can be reached by application of risk priority numbers, RPN, displaying specific values for each risk attribute. Here is the motto: the more in-process controls are implemented at critical phases in development, production, or application, the more discoverability is given. While severity is not directly capable of being influenced, risk probability can be managed by application of high-grade material and regular training of professionals to keep RPN numbers for probability low. For performing risk management, whole life cycle of medical products have to be investigated. Being part of technical documentation, risk treatment is compiled within a risk management file. Decisions considering risk management are made by representatives from different departments, for example, development, production, marketing, technical support, and executives. These delegates unite in one risk management team, meeting regularly in the range of 12 months and also separately in case of appraisal of reclamations. In the following, templates for documentation of risk parameters will be illustrated. Every hazard, chemical, production, application, or information consists of different potential mistakes coming from defined objects. These objects are involved in mistake scenario. What is the potential mistake? What is the potential consequence? And what is the potential mistake reason? Every scenario is receiving individual control directives announced by a risk management team, leading to new current status after risk management. This status has to be evaluated then by risk priority numbers. The object here is on the one hand upstream processing and on the other hand downstream processing. In both cases, there may be insufficient control of production processes or control of changes in the production process. The potential consequence could be that in the final product there is a false negative result in the applied ELISA assay. The potential mistake reason could have been that a recombinant agent which has been produced in manufacturing got denaturated in cell culture, so for upstream processing, or during chromatography during downstream processing. During manufacturing processes of recombinant proteins, numerous in-process controls can be implemented to keep risk priority numbers for discoverability at low values.